Collagen is one of the most important structural proteins in our body. It acts like a framework that gives our bones, skins, and tendons their strength and flexibility. This is normal collagen. It's made up of three chains that twist together into a strong triple helix. Every third amino acid in this chain is glycine, which allows the chains to pack tightly together. Glycine is the smallest amino acid, and its side chain is a single hydrogen atom, making it nonpolar and uncharged. This is perfect for fitting inside the tight spaces of the helix, which keeps the collagen flexible and stable through hydrogen bonds. Under normal conditions, collagen is made inside the cell as a molecule called procollagen. It's formed under the three protein chains that twist together into the strong triple helix. It's the helix shape that provides the stability and strength for our connective tissues. Collagen synthesis begins in the ER, where the chains are assembled and then folded. Then the procollagen is then transported through the Golgi apparatus and sent out through the cell through exocytosis. Once outside, special enzymes work to convert procollagen into mature collagen fibers that assemble into strong fibrous networks. Collagen is primarily located outside of the cell in the extracellular mix, where it forms fiber networks and scaffolds to support tissues and organisms. Collagen is categorized as a structural protein because its main job is to provide mechanical support and shape to the body. There are several types of cells that produce collagen, each for specialized different tissues. For example, fibroblasts make collagen in connective tissues for like skin and tendons. Osteoblasts make collagen in the bone. Chondrocytes make collagen in our cartilage. And now, imagine there's a mutation that replaces glycine with a different larger amino acid like cysteine or serine. That small change may not seem like much, but a larger polar serine can't fit inside a tight collagen helix. This causes crowding and forms abnormal hydrogen bonds and disrupts the perfect twisting of these three chains. The triple helix becomes unstable and weaker. Inside the cell, the misfolded collagen gets stuck in the rough ER, and the buildup of faulty proteins stresses the cell and can cause cell damage. The collagen that does get secreted into the body forms weak, improperly cross-linked fibers. This first model represents normal collagen, which has two alpha 1 chains and one alpha 2 chains. These chains are made up of glycine, which allows the chains to pack tightly together, giving them a strong, stable, and flexible structure that helps to provide strength to bones and mutated collagen. As you can see, at the point of mutation, one of the glycine amino acids is replaced with a larger amino acid like cysteine, and this results in a disruption of the triple helix. Because serine is larger and more reactive, it doesn't fit smoothly where the glycine used to be. This creates a steric hindrance, meaning the side chains bump into each other and our triple helix will become unstable and the hydrogen bonds will be disrupted. As a result, the collagen fiber weakens and cannot maintain its structure. The substitution of glycine within this triple helix disrupts the precise alignment of the chains, resulting in a weaker and more unstable structure as compared to the traditional collagen. While the models used effectively demonstrate the difference between normal and mutated collagen, it's important to consider the limitations that differentiate a visual tool and the true biological structure of the protein. The conventional model lacks the depth and visuals of the amino acid sequences and the alpha helix structures. It's also inaccurate and the proportions are not made to 3D models serve as a valuable visual tool to understand how a single amino acid change can significantly impact protein structure and biological function. It's important to remember that the 3D models only show a visual difference in the collagen and not how it affects bone strength as it relates. How does this relate to the world of medicine? I'd like to introduce you to osteogenesis imperfecta, or OI, which is a genetic disorder that makes bones fragile and easy to break, giving it the more common nickname brittle bone syndrome. In people with OI, there are dominant mutations in the COL1A1 or the COL1A2 genes. These genes are responsible for carrying the instructions for making the collagen protein. When they mutate, the helix can't form correctly. Without properly folded collagen, these tissues will lose their stability, leading to symptoms of osteogenesis imperfecta.